You know, usually when we talk about a severe injury, like a deep laceration or a surgical wound or, a third-degree burn, there is this baked-in expectation that a scar is just, well, it's the inevitable price of survival.
Right, yeah. It's just a given.
Exactly. It's like your body is pulling the emergency brake on a train. It stops the bleeding. It seals the gap, which prevents fatal infection, obviously. But the original track is just permanently deformed.
Yeah, it's a rapid functional patch job. Biologically speaking, we trade structural integrity and complex function for speed and immediate survival.
But what if that emergency brake isn't there because the body actually can't rebuild the track? What if the exact biological blueprint to perfectly regenerate complex tissue is still just sitting dormant inside you?
Like it never actually left.
Right. But a hyperactive alarm system in your own body is actively hitting the stop button. So welcome to this deep dive. Today, we are exploring a genuinely mind-blowing 2026 paper from the journal Cell, published by Tam and colleagues.
It really is paradigm shifting, yeah.
Yeah, it totally is. And our mission today is to completely flip your understanding of why mammals lose the ability to regenerate complex organs. We're going to look at the exact molecular mechanisms that might allow us to, switch that latent ability back on.
And to really grasp just how groundbreaking this research is, we first need to establish a hard line between two distinct biological concepts. So, tissue-level regeneration and multi-lineage or organ-level regeneration.
Okay, right. Let's make sure we define those.
Yeah, so tissue-level regeneration is something your body does constantly, like you scrape your knee and the superficial epithelial layer turns over and replaces itself. It's essentially just a single-cell lineage proliferating to fill a shallow deficit.
Which is relatively simple, right? But multi-lineage regeneration is an entirely different beast. I mean, mammalian skin is a deeply complex, multi-layered organ.
Oh, absolutely.
You're talking about hair follicles, sweat glands, dermal adipocytes, which are fat cells, robust vascular networks, and these highly intricate sensory nerve architectures. Rebuilding all of that from scratch, coordinating multiple distinct stem cell lineages to recreate the exact correct spatial architecture.
That is true organ-level regeneration.
Right. And in adult mammals, it is exceedingly rare. Okay, let's unpack this, because the foundational premise of this paper is that we actually do possess this multi-lineage superpower, but we lose it incredibly early in life.
Yeah, the researchers set up this baseline experiment comparing the healing capabilities of mice at two distinct developmental stages. They looked at embryonic day 16.5.
Yeah.
So late-stage fetuses still developing in the womb.
Okay.
And postnatal day 5, which is just 5 days after they're born.
And the structural contrast between those two stages is just staggering.
It's night and day. When they created full-thickness excisional wounds in the embryonic mice, the skin healed flawlessly. No fibrosis, no scarring at all.
Mm-hmm.
If you analyze the collagen extracellular matrix under a microscope, it formed this healthy interwoven basket weave pattern, which is the exact architectural signature of normal, uninjured skin.
And crucially, it wasn't just the extracellular matrix returning to normal. The appendages came back. The hair follicles regenerated, the dermal adipocyte layers reformed, the erector pili muscles regenerated.
A complete restoration.
But then you look at the cohort wounded just 5 days after birth. That regenerative superpower is completely abolished. The skin forms a dense fibrotic scar characterized by these rigid, parallel bundles of collagen. The complex dermal appendages they fail to form entirely.
Exactly. It just defaults to a patch.
But wait, couldn't this just be an environmental factor? I mean, the amniotic environment is completely sterile and bathed in growth factors. Once a mouse pup is born, it's immediately exposed to air, mechanical stress, an onslaught of external bacteria.
Right. The environment totally changes.
Yeah, which naturally alters the inflammatory baseline of the tissue. So are we sure the innate biology is actually broken? Or is the newborn just dealing with a dramatically harsher environment that forces a rapid scar response?
See, that is exactly the variable you have to isolate. And the researchers tested the environmental hypothesis really rigorously. They wounded a cohort of embryonic mice at day 16.5, but then they manually delivered those pups early at day 18.
Oh, wow. So they took them out of the womb.
Right. This meant the pups spent the vast majority of their critical healing window outside the uterus, fully exposed to that exact harsh, non-sterile external environment you just mentioned.
And the result, did they scar?
They still regenerated perfectly.
That is crazy.
Yeah, the sterile uterine environment isn't the magic ingredient driving the healing. Something fundamental inside the intrinsic biology of the skin itself drastically shifts right around the time of birth.
Okay, but what if postnatal healing is just like slower or delayed due to all the metabolic demands of being a newborn?
They ruled that out too. They use high-resolution 3D whole-mount imaging to track those postnatal wounds for 50 days. In neuron development, 50 days is a massive window.
Right.
And the scar remained permanent. The regenerative program wasn't just delayed, it was fundamentally locked out.
To prove just how flawlessly that embryonic skin was regenerating, the researchers utilized a physiological marker that I found absolutely fascinating, the piloerection reflex, essentially the goosebump test.
Oh, I love this part. It's such an elegant way to prove function.
Right. For a mammal to physically get a goosebump, a highly specific trilineage unit has to form and wire together perfectly. You need the epithelial hair follicle, the mesenchymal erector pili muscle, and a sympathetic nerve fiber that correctly synapses onto that muscle to trigger the contraction.
Exactly. It requires the flawless spatiotemporal coordination of 3 completely different cell types.
Yeah. And when the researchers placed these mice, the ones that had completely healed from the embryonic wounds, into a cold chamber at 4 degrees Celsius, the regenerated wound bed reacted instantly. The skin got goosebumps.
Which is just incredible.
It really is. That confirms the body didn't just throw some undifferentiated cells into the deficit to plug the hole. It perfectly orchestrated the reconstruction and wiring of a functional multi-tissue organ from scratch.
So let's do a quick recap here. Bullet 1, embryonic mammals can perfectly regenerate complex skin organs, complete with hair and functional nerves.
And bullet 2. This ability vanishes within days of birth, replaced entirely by permanent scarring.
Right. Which leads to the big question for you, the listener. If the exact genetic and cellular blueprint to rebuild complex skin exists and works perfectly at day 16, where does that blueprint go by day 5 after birth?
Yeah, why does the system suddenly default to a rigid scar?
To answer that, the researchers had to map the cellular landscape at an incredibly granular level. They knew this transition from perfect regeneration to scarring happened rapidly, so they utilized single-cell RNA sequencing on the wound beds just 3 days post-injury.
Which is so cool because that allows you to look at the entire population of cells in the wound, read their individual transcriptomes, and see exactly which genes are actively being transcribed in real time.
Precisely. They were essentially looking for the biological variable that was present in the postnatal scar tissue, but absent in the perfectly healing embryonic tissue.
The smoking gun, basically.
Yeah. And while they noted some expected shifts, like an increase in certain immune myeloid lineages in the postnatal wounds, the absolute standout discovery was the emergence of a highly specific transient subpopulation of cells. They dubbed them postnatal wound-specific fibroblasts, or PWFs.
Here's where it gets really interesting, because historically we've thought of fibroblasts primarily as just structural worker bees. They just synthesize collagen.
Just laying down the bricks, yeah.
Right. But this highlights the incredible heterogeneity of fibroblasts. These specific PWFs are practically non-existent in healthy, uninjured tissue. They are completely absent in the perfectly healing embryos.
Completely absent.
But the moment a postnatal mouse sustains a skin injury, these PWFs rapidly expand. They dominate the wound bed to the point where they make up about 60% of all fibroblasts present within just a few days.
They completely hijack the cellular environment. And when the researchers analyzed the specific transcriptome of these PWFs, they found they weren't just structural, they were highly enriched for genes encoding secreted factors.
Meaning they're sending out signals.
Exactly. These cells act as powerful signaling hubs, constantly pumping out chemokines and signaling proteins into the surrounding extracellular space.
But correlation isn't causation. Just finding these unique fibroblasts at the scene of the postnatal scar doesn't guarantee they're the ones pulling the emergency brake on regeneration.
Which is why the experimental design here is so elegant. They performed a highly targeted in vivo functional screen. They isolated 9 of the top secreted factors produced by these postnatal PWS.
And their hypothesis was, if one of these chemical signals is the true brake pedal for regeneration, artificially introducing it into an embryo should override the embryo's natural regenerative superpower.
Right, it should force the embryo to form a scar.
So to do this, they utilized adeno-associated virus vectors, AAVs, which you can think of AAVs as basically microscopic delivery trucks. They use these trucks to selectively overexpress these 9 postnatal secreted factors directly into the embryonic wound beds.
So they were essentially trying to infect a perfectly healing embryo with the postnatal scarring instructions.
Yeah, exactly. So what happened?
Well, they tested all 9 candidates independently. 6 of the vectors had no negative effect. The embryo simply ignored the signal and healed perfectly.
Okay, so 6 duds.
Right. But 3 specific factors successfully broke the regenerative program. When they overexpressed the genes CCL7, CSCL12, or TIMP1, the embryonic wound stopped regenerating.
Wow.
Hair follicle meiogenesis failed. Dermal adipocytes failed to form. The sympathetic nerve networks collapsed. The tissue transitioned to a fibrotic scar.
So they successfully transferred the postnatal scarring phenotype into the embryos. Think about the implications of that for a second. The loss of regeneration after birth isn't just a passive degradation of stem cell capacity.
It is an active suppression.
Yes. The body is spawning these PWF cells, and those cells are actively secreting factors, specifically CXCL12, CCL7, and TIMP1, that chemically sabotage the regenerative blueprint.
Which logically demands we ask the next mechanistic question, right?
Right. Time for another quick recap. Bullet 1. Postnatal wounds spawn a unique cell, the PWF. Bullet 2. PWFs secrete specific factors like CXCL12 that actively block regeneration when introduced to embryos.
So the question for you listening is, how exactly are these 3 chemicals sabotaging the healing process? Are they directly poisoning the stem cell niches? Are they forcing early differentiation?
This was the twist in the paper that genuinely surprised me, because when you look at the downstream effects of those 3 guilty genes, they share a very distinct, defining characteristic. When introduced into the embryonic wounds, all 3 heavily promoted a state of hyperinnervation.
Hyperinnervation is exactly what the prefix implies. It's an excessive, chaotic, and overly dense sprouting of sensory nerve fibers aggressively invading the injury site.
Too many nerves, basically.
Way too many. And of the 3 specific factors, CXCL12 was identified as the primary driver of this phenomenon. CXCL12 is a potent chemokine, and its specific binding partner is the CXCL4 receptor, which is highly expressed on the surface of sensory neurons.
So they lock together.
Right. In the context of the wound, the massive release of CXCL12 from the PWFs acts like an overwhelming chemical magnet, aggressively yanking dense tangles of sensory axons right into the center of the granulating tissue.
Hang on, I want to look closely at this, because everything we are taught about the pathology of scarring and fibrosis points to inflammation as the primary culprit.
Oh, absolutely. That's the classic dogma.
Right. The established dogma is that overactive macrophages, neutrophils, and an aggressive cytokine storm are what drive the fibrotic response and prevent complex tissue regeneration. Are we absolutely certain is this neural hyperinnervation blocking the healing process? And not just the fact that these chemokines are simultaneously calling in a massive influx of immune cells?
It is the most critical confounding variable to address because, like you said, it challenges decades of assumed fibrotic biology.
So how did they prove it?
To definitively decouple inflammation from hyperinnervation, the researchers intentionally induced massive, overwhelming immune responses in the embryonic wounds to see if severe inflammation alone could break the regenerative capacity.
Oh, that's clever.
Yeah. First, they injected LPS glycopolysaccharide, which is a highly immunogenic component of bacterial cell walls, directly into the amniotic fluid to simulate a severe infection model.
So they created a highly toxic, highly inflammatory environment.
Exactly. The wound beds immediately flooded with myeloid lineages and acute inflammatory cells, but the embryonic skin still regenerated perfectly with full appendage formation.
It just ignored the inflammation.
Completely. And they didn't stop at the LPS model. They utilized the AZ vectors again, this time to overexpress massive immune-boosting cytokines like CXCL5 and CSF2 directly into the embryonic wounds.
Forcing another massive influx of inflammatory cells.
Right. And once again, the tissue achieved perfect multi-lineage regeneration. The sheer volume of inflammation or immune infiltration does not block organ-level regeneration.
Wow.
But every single time they induce that chaotic sensory nerve overgrowth, that hyperinnervation, the regenerative program crashed into a fibrotic scar.
Okay, let me try an analogy here to visualize this mechanism for everyone listening. Think of the wound bed like a garden where you are trying to cultivate a highly complex ecosystem of specific plants. Those are your regenerating hair follicles, your sweat glands, your fat cells.
Like this.
Right. So the long-standing medical assumption was that a bad scar means the soil itself is toxic, that severe inflammation is poisoning the seeds. But this paper proves the soil is actually fine. The problem is that the PWF cells act as a powerful selective fertilizer for vines.
Oh, that's a perfect way to picture it.
Yeah, these sensory nerves grow so incredibly fast and so densely that they physically and chemically choke out the entire garden.
That analogy holds up perfectly on both a structural and biochemical level. Because these are raw, highly active, nascent nerve fibers, they are incredibly excitable. They are constantly leaking excessive neuropeptides and neurotransmitters into the microenvironment.
So it's just chaotic chemical noise?
Exactly. That erratic, high-volume neurosignaling completely scrambles the delicate morphogen gradients. Those are the precise spatial and chemical signals that the stem cells desperately need to organize into a hair follicle or a complex gland.
So the paradigm shifts completely here. For you listening, this is the massive takeaway. So recap time. Bullet 1, hyperinnervation. Too many nerves blocks regeneration, not immune cell inflammation.
Bullet 2, the PWF cells are acting like a beacon, calling in a tangled mess of nerve fibers.
Which brings up our next question. If the nerves are the roadblock, what happens if we simply clear the road? Can we therapeutically decouple that interaction and trick postnatal skin into healing like embryonic skin?
Right. If the nerves are blinding the stem cells, how do we shut off the beacon? They approach this through two distinct avenues: genetic manipulation and pharmacological intervention.
Let's start with the genetics.
Okay, so genetically they utilize conditional knockout mouse models. In one cohort, they specifically deleted the CXCL12 gene strictly from the fibroblast populations. In a separate cohort, they deleted the CXCR4 receptor strictly from the sensory nerves.
Okay, so in both isolated models, they successfully broke that magnetic attraction.
Exactly. The sensory nerves were essentially rendered blind to the postnatal wound environment and didn't aggressively invade the tissue.
So you prevent the hyperinnervation from happening in the first place. And when they wounded those postnatal day 5 mice with the genetic knockouts...
Perfect regeneration.
That is wild.
The postnatal skin, which should have scarred, healed flawlessly. The hair follicles, the dermal fat layers, the erector pili muscles, all of it regenerated. The normal, organized neural networks were formed without the chaotic overgrowth. By simply lifting that specific neural blockade, the innate, latent regenerative superpower of the skin was completely reactivated.
But clinically speaking, we can't easily perform a conditional genetic knockout on a human patient who just presented to the ER with a 3rd-degree burn. We need a translatable therapeutic.
Right, something we can actually give to a patient.
And this is where the paper offers a pharmacological application that is just brilliant. They utilized botulinum neurotoxin A, widely known as Botox.
It sounds like a cosmetic gimmick at first glance, I know. But the molecular mechanism is incredibly precise and perfectly suited for this exact problem. Botox doesn't cause apoptosis. It doesn't kill the sensory nerves. It specifically silences their synaptic transmission.
Let's get into the biochemistry of that for a second because it's really important. Botox acts as a highly specific protease. It enters the nerve terminal and cleaves a protein called SNAP25.
Right.
And SNAP25 is a critical component of the SNARE complex. The SNARE complex is essentially the molecular machinery that allows synaptic vesicles, the little bubbles full of neurotransmitters, to fuse with the presynaptic membrane and dump their chemical payload into the surrounding tissue.
Exactly. So if you use Botox to cleave SNAP25 and destroy the SNARE complex, the nerve fiber remains structurally intact, but it is functionally muted.
It is physically incapable of releasing those disruptive neuropeptides into the wound microenvironment.
It silences the chaotic chatter. So the researchers took wild-type postnatal mice, no genetic modifications at all, and simply injected botulinum neurotoxin A directly into the wound bed.
And
by chemically quieting that excessive nerve chatter, multi-lineage regeneration was massively restored.
Just from Botox.
Yeah. But the detail that really highlights the translational potential here is that they didn't just test this on 5-day-old neonatal mice. They tested the exact same Botox intervention on fully grown adult mice.
Oh, wow.
And it significantly enhanced complex appendage regeneration in the adult tissue as well, shifting the healing process away from a flat fibrotic scar and back toward functional architecture.
If we connect this specific molecular pathway back to the broader evolutionary picture, you really have to ask why mammalian biology evolved this specific mechanism. I mean, why would natural selection intentionally favor the development of a cellular alarm system that actively suppresses our ability to heal perfectly?
It seems completely counterintuitive, right? Like a massive biological design flaw.
Yeah, exactly.
But it's not a flaw. It is a brutal, calculated evolutionary trade-off. Think about it. When a baby mammal leaves the womb, it instantly enters an environment fraught with predators, mechanical hazards, and massive infectious risks.
It's a dangerous world.
Very. It urgently requires a highly calibrated, hypersensitive alarm system to immediately register pain, to learn to avoid noxious stimuli, and to survive its most vulnerable developmental window. That rapid, dense sprouting of nascent sensory nerves in a fresh wound drives acute hypersensitivity.
Ah, so if a wound is excruciatingly sensitive, the animal will aggressively guard it.
Exactly, which prevents subsequent mechanical damage or fatal infection.
So it's prioritizing immediate survival over long-term perfection.
Precisely. We permanently trade our slow, meticulously perfect regenerative capacity in exchange for a rapid response, hypersensitive fibrotic patch that ensures we actually live to see tomorrow.
That makes total sense.
But what this paper demonstrates is that the old embryonic blueprints aren't destroyed in that trade-off. They're just buried under the noise of the alarm system.
So our final mini recap here. Bullet 1. Blocking the chemical signal, CXCL12, stops the nerve overgrowth. Bullet 2. Silencing the nerves with Bonte Botox unlocks the skin's latent ability to regenerate, even in adults. By therapeutically intervening, we can essentially buy the stem cell niches a quiet, localized window of time to access those embryonic instructions and fully rebuild the tissue.
It completely shifts the therapeutic paradigm.
So what does this all mean for you listening? Think about the future of medicine. The implications here are just profound. Think about the current standard of care for chronic diabetic ulcers, severe surgical resections, or massive burns.
It's mostly just managing the scarring.
Right. For decades, the entire field of regenerative medicine has been heavily focused on trying to engineer external solutions. Trying to bioprint new skin or flood a wound with exogenous stem cells to force the body to build complex tissue.
We've been trying to invent a brand new healing machine from the outside in.
Exactly. But this research proves that we don't need to build a new machine. The machinery is already inside us. It has been sitting there perfectly functional since before we were born. We don't need to push the gas pedal harder. We just need to figure out how to take our foot off the brake.
And it leaves us with a highly provocative, almost unsettling biological question to end on.
I love these. Let's hear it.
In this deep dive, we've clearly seen that mammalian skin holds a dormant, suppressed superpower to perfectly regenerate. And that ability is locked away by the aggressive hyperinnervation of our own sensory nervous system.
Right.
It forces you to look at the rest of our anatomy and wonder, what other critical non-regenerating organs in the human body, say, myocardium that has suffered an infarction, or central nervous tissue severed by a spinal cord injury, or even entire lost limbs, might actually possess those exact same hidden blueprints.
Oh, wow.
What if the ability for a human heart or a human spine to perfectly rebuild itself is already there, perfectly intact, if we could only figure out which specific biological alarm system is standing in its way?
It's like staring at a train that's been stopped on the tracks for our entire lives. We always assume the engine was dead or the tracks were destroyed, but maybe the infrastructure is perfectly fine. Maybe we just need to learn how to let go of the emergency lever.